Thank you. Hello everyone, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati. And this is the first live session of the July 2023 course. So if you have any queries, you can ask me the questions. This is a very, this is going to be a short live session, depending upon the number of applicants going to be present and ask me the questions. So if you have any queries, please post your questions so that I will be happy to answer them. Oh, okay. Fine. So we have our first query that is from Chinmaya, and the Chinmaya and has asked about the format of the exam, right? So the format is already been given on our website. So it is going to be a multiple choice questions where we are going to give you the 100 questions, and the time allotted for completing this these questions is 3 hours, and there's no negative marking, so you can attempt all the questions, and the passing marks and other things are already been elaborated on our website. So passing marks would be based on your performance in the assignments, and as well as your performance in the final exam. So you have to do very good in terms of in the assignments and as well as in the final exam to be eligible for getting a certificate. Currently, we are not giving any hard copy certificates. So all the soft certificates you are going to get in the soft copy. And so this is just the basic information about all the questions, all the queries which is related to the pattern of the question papers and the type of question papers. Apart from the numbers, the pattern would be that we are going to have at least 25 to 30% questions from the assignment type, and few questions would be of theory based, and few questions would be experimental based. So I think these are the things what you can actually be able to be able to answer. And I have prepared a question paper in such a way that if you have gone through the content, I think you will be able to answer all the questions and you will be able to gain good marks. So if you have not registered for the final exams, you can actually be able to register, and I hope the course is going to be a good source of information for you and you will be having a good learning experience. So if you have any more questions, you can ask me. So Chinmaya, I think hopefully you got the answer, and others also. So it's going to be a 100 question MCQ type with no negative marking, and the time allowed is 3 hours. Now we have another question from the Zara. She is saying that, sir, when we will going to get the answers of the assignment? So once the due date of the assignment is over, then you are going to see you can be able to see the answers of your assignments, and you can be able to you know, you will even that time only it is actually going to display the marks for you also. The Chinmay is also asking, please explain the counting calculation in hemocytometer. So calculation is very simple actually. So hemocytometer, the uh, the 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 volume of the each chamber, what you are actually going to get is approximately 20 microliter. So you can actually be able to count the number of cells in 4 different chambers, right? Remember when the Banish was showing you how to count, and then you can imagine that each chamber is actually 20 microliter, right? So if these many number of cells are there in 20 microliter, what will be the number in 1000 microliter? So it is a simple math, and you can be able to do it. Just for making it more statistically significant, you are supposed to count the cells in all the 4 chambers, because depending upon the type of distribution, the cell number would be more in one chamber compared to the another chamber. So it's important that you should count the cells in all the 4 chambers, and then whatever the number you get, you divide that by 4. So these many number of cells are present in the 20 microliter. So you can calculate with the simple mathematical formula what will be the number in the 1000 microliter. I hope it is clear now. Inmaya, is it clear now? So there is no, Chinmaya is having another question. So there's no negative marking in the assignments or even in the final exam also. So you can be able to attempt all the questions, and hopefully you will be able to get good marks. And most of the assignments are from the content only. So if you go through with all the lectures and the demo videos, I don't see any question which you cannot be able to answer. If you have any more questions, you can ask me. Uh, so Zara is asking about genomic library preparation, I think. So genomic library preparation is very simple. Uh, so what we are going to do is, uh, wait, I'll just see whether we can be able to write. Huh? इसमें display कैसे है? one, right? Correct. Thank you. So, so what we are going to do is, first of all, in the step one, so in the step one, you are actually going to isolate the genomic DNA, right? And once you isolated the genomic DNA, you are actually going to fraction you are going to make the fragments. Okay, and I think we discussed several methods. So one of the method is you can going to treat the genomic DNA with the restriction enzyme. The second method could be that you are just going to do the mechanical fragmentations. And the third is you can actually be able to do the sonication. Whichever the method you want to use, once you got the fragments, you are going to get the small fragments, right? Now, each of these small fragments, you are actually going to put it into a vector, which is going to harbor this particular fragment. So if you put the, if you take the vector also and digest it with the restriction enzyme, it is actually going to take up all these fragments. So that's why you are going to have the a collection of the clones, collection of these fragments, right? So you can have this into this one into this one, like that. Okay. And then what you're going to do is in the step 3, you are going to take this and put it into a transformation. Okay. So once you do the transformation, you are going to get the colonies of the particular bacteria or, and these colonies are nothing but the genomic library. So it's going to represent the genome, but the individual gene is actually going to be present in the individual bacterial colony. So if I want to isolate a particular gene, what I can do is I can just screen this genomic library with the help of the either the DNA probe or with the help of the antibody based method, or we can actually be able to use the enzymatic method, and then it is actually going to say that, okay, this is a clone, what is actually showing me the positive signal. And then what I can do is I can isolate this particular clone from my master colony. So this is just a simple and a very quick overview of how you can be able to use the how you can be able to understand the genomic library. I can use the different colors, but I am not that good actually in terms of changing the color, right? Compared to the things. So Zara, I think you are misunderstood that in the genomic library we have all same type of genes. No, you're not. Genomic library is just like your books library. In books, you in a normal library, what you have in a normal library, you have the books of different subjects. You have the books of the different authors. Similarly, you're in the genomic library also. You're going to have the fragments. These fragments, right? With color. For example, this is the suppose this is the fragment what you have in the yellow one, right? Suppose this is the fragment which is coding for actin gene. Okay. Then you can also have another fragments like green, for example. So this the green DNA, what you have is actually a fragment which is which is coding for myosin. I'm sure you are familiar with these names. Okay. So actin is another gene, myosin is another protein. Our muscles are made up of actin and myosin and so on. So if once you clone, some of the, so for example, this one, this clone is probably will have the myosin. This clone will actually going to have the actin. This clone will have the hemoglobin. This clone will have LDH. This clone will have something else. So every clone is actually going to have the individual genes, and that's how you can be able to isolate and select the gene of your interest, because you can use the DNA probe for isolating the actin. So when you use the DNA probe for actin, it is actually going to say that, okay, I'm interacting with this one. Okay, this means this is the clone, or this is the bacterial. This bacterial colony actually contains a clone which has the actin gene. It could be fully gene, it could be full gene, or it could be a partial gene. But many of the clones will actually going to have this particular gene. So you can be able to isolate the gene from these libraries. And that is what the purpose of having the genomic library. I'm happy that you understand, and I hope it will help you in in advancing your study. So there is another question from Chinmaya that can I show the certificate in the PhD interview? So definitely you can be able to show the NPTEL certificate in any of the interviews and in your job interviews and PhD interviews and other places. It is internationally valid, so you can be able to use that for wherever you require. And there's no authorization or anything required. You can actually be able to freely use this certificate, and that is what the advantage is, that you actually make your CV more rich by going through these kind of online courses, and that's how you can be able to learn the new techniques and learn the new skills, and at the same time you can be able to continue with your work what you're doing now. So if we have any more questions, please post your questions. Otherwise, we will wind up in another few minutes. If there will be no question, then we will wind up quick, sooner than the 1 hour actually. I hope Zara and Chinmay, both you got the answers and you are fully satisfied. So I enjoyed interacting with both of you, and I hope that all your doubts are clear, and actually it will help you to clear some more assignments in the coming week. So with this, I think I would like to conclude my live session today. And in our subsequent live session, we are going to discuss some more problems from you, because by the time you will be even more in an advanced stage, so you will actually be able to understand the subject more thoroughly, and at that time probably we'll have more interactive sessions. So with this, I would like to conclude my session here. In our subsequent session, which is going to be the next month, we will have a more discussion about the subjects. Thank you. Kundavan,